Hey, our topic is gene therapy, CRISPR-Cas9, and how it can be utilized to treat DMD, Duchenne muscular dystrophy. So first off, let us explain what DMD is, how it affects people, and how scientists are trying to find a treatment.
DMD, or Duchenne muscular dystrophy, is a fatal genetic disease which is caused by a duplication in a human's genetic code, or their DNA. The primary symptom of DMD is muscle weakness, which starts around the age 3. This will become more severe over time, eventually affecting muscles such as the heart and lungs, which can in turn be fatal.
Now, to give you some background information on DNA. DNA is the code made up of different bases which can be read in order to produce RNA, which in turn can be used to synthesize a protein. As you've started to learn, these proteins help determine our physical characteristics. Mistakes in DNA can in turn create non-functional or partially functional proteins, causing problems in the way your body works.
These mistakes or problems in the DNA are called mutations. DMD is the result of a mutation in a specific region of the DNA code, which codes for the dystrophin protein. This protein stabilizes muscle cells, and without it, causes degradation or dystrophy of muscle cells over time. The specific type of mutation occurring is a frame shift and results in a non-functional dystrophin protein.
The code of DNA is translated in three base pair segments known as codons, each coding for one amino acid of the protein. If a deletion, addition, or duplication occurs which is not a multiple of 3, the reading frame of the DNA sequence is shifted, leading to a change in the amino acids in the protein, in many cases also causing a non-functional protein to be produced. This is the case with DMD.
Now, I know you've been learning about DNA and DNA technology, and the method we're going to explain to you today is a very interesting, useful, and new technology for altering specific DNA sequences known as the CRISPR-Cas9 system.
Now, CRISPR-Cas9 is a gene editing system scientists adapted from learning about bacterial immune systems. The CRISPR-Cas9 system is able to make targeted edits in the genome. This is accomplished using what is called a guide RNA and a protein called Cas9. The guide RNA is made to be a complement of the DNA, being targeted for edits, and will bind to the matching sequence. The Cas9 protein is an enzyme that will cut both strands of the DNA and is directed by the guide RNA. Once the Cas9 makes the cut in the genome, the cell will try to fix the break. This can cause a deletion or insertion of random base pairs, which can alter the reading frame, disrupting the function of a gene, as discussed earlier.
We can use this system to edit the dysfunctional dystrophin gene. We can direct the Cas9 enzyme to cut the DNA in a way that corrects the reading frame. This does not make the normal dystrophin protein, but rather a stable and functional protein that is similar to the normal dystrophin gene protein. This protein will act like dystrophin and cause muscles to function much more normally than if the dystrophin had the non-functional reading frame mutation.
After we've created this new dystrophin-like protein in stem cells in culture, they are then transplanted into the skeletal muscles of the patient. This process was first done in mice in order to test for the efficiency of the system and later in human trials. This method yielded fairly successful results, which gives rise to new research that will assist in treating other genetic disorders in the future.